Thanks, Crawford. Thanks, everyone. So most of my research has really been focusing on this intersection between these three intrinsically linked processes. And this area has received a lot of attention on land with terrestrial agriculture, but there's been significantly less focus paid in the water, with the one exception, as we can probably all appreciate, being salmon lice in salmon aquaculture. And as we can probably appreciate as well, the evolution of sea lice in response to the new pressures found in salmon aquaculture has significant implications for the immediate industry. But I also think that looking at sea lice evolution can actually be a really interesting case study into help informing us how pest management in aquaculture more broadly can be coordinated into the future. So there are 5 main groups of chemicals that have been used in Norway, especially in salmon farming over the last 30 or so years. This is the introduction in Norway. And resistance has evolved in sea lice to 4 of these 5 main chemicals. These arrows indicate the first experimental evidence of resistance in sea lice in the Atlantic Ocean. And as we can see, the time frame is really, really short. So lice have evolved resistance in a really, really short period of time. And as resistance becomes more widespread, the efficacy of these chemicals decreases. And so the use of these chemicals have actually plummeted in Norway over the last 5 or so years. And so I'm going to be talking about the evolution of treatment resistance in sea lice. And when I talk about evolution, I'm talking about microevolution, which is the change in gene frequencies over time in a population in response to a selection pressure, in this case, delousing treatments. And this shouldn't have come as a surprise that occurred in sea lice because it's been seen time and time again in terrestrial agriculture. But this means we can also learn from what we see on land and find new ways to prevent risk of resistance from arising in the first place. This is rather than simply waiting for it to arise and then relying on new treatment technologies to take their place in the future. And to do this, we can apply evolutionary principles into how we actually manage treatments or other control strategies on salmon farms. So integrated pest management, it's a well-known term in terrestrial farming. And there are a number of different ways that we can coordinate treatments to slow or prevent the spread of treatment resistance in sea lice. We can combine multiple treatments through space or through time. And this effectively attacks the lice from multiple different angles and makes it harder for them to develop resistance to the suite of strategies available. Another option is to leave treatment-free refugia. So these refugia support a population of susceptible lice. When these transfer into treated farms, they effectively dilute the genetic resistance on those farms. And of course, another option is to turn to non-chemical alternatives. For example, we have mechanical delousing, cleaner fish, lice skirts. Something we might hear about a bit more over the next few days is the use of new gene editing technologies to change the salmon themselves so they are themselves more resistant to salmon louse infection. So all these non-chemical methods obviously avoid the issue of pesticide resistance, but there is still concern that these can impose selection pressures on the louse population in different ways, and lice could evolve resistance in some capacity to these new treatments, which is something that I've explored a little bit in this review paper. And the take-home message is that there's no one-size-fits-all approach to pest control in any system. What we need to do is find the optimal management strategy for any system that firstly limits the evolution of resistance to that strategy, but at the same time still manages to keep the pest under control in the short term. And to find this optimum balance, we can turn to evolutionary models, metapopulation models. As we've seen already today, there's been some fantastic population models that have been developed and used through time. There's also been some really good evolutionary models looking at how sea lice populations evolve in response to treatment pressures. So what I'm talking about today is my attempt to integrate these 2 components into one model that looks at how sea lice might be able to evolve across a farm network. So the first thing I'm going to do is just talk about how I went about constructing such a model. I'm going to avoid going to the equations, parameters used, and just look at it from a more conceptual approach here. So the first component is developing that population model that tracks individual lice through time on individual farms. So here's the lice infecting salmon. They go through the life stages from calamus to the adults. The adults produce new larvae, and we have a background mortality rate as well of these life stages. The model also includes a component that allows for delousing treatments. So for example, if the number of adults exceed a certain threshold, a treatment is applied and additional mortality occurs for the lice. And we use the Norwegian legally mandated lice limits for this model. We can also assign a farm some sort of preventative or continuous louse management strategy, for example, cleaner fish. If there are cleaner fish on the farm, there's additional mortality of the lice. Or we could play around with hypothetical new methods of louse management, for example, some sort of future resistant salmon strain that affects the survival of lice at different stages or might affect the fecundity of the adult lice. So we can really use all sorts of different imaginary or real louse management strategies in these models. Of course, we need to complete the life cycle, so we need to figure out how many larvae produced at the farm end up returning to that farm and reinfesting it. This is where we turn to the sea lice dispersal model, which, as Trondor has already discussed a little bit, is this really powerful tool that we can use to track how individual lice particles are transported through the ocean environment on ocean currents. And here's a little simulation again of that model in action. So we can place our farm in our model here, for example, and figure out how many of the larvae produced at any point in time end up returning to that farm. Or we can estimate how many lice might transport to another farm here, for example. So that's the first part of the model, the population dynamics part of the model at individual farm sites. We now want to start looking at the evolutionary dynamics involved. And for this, I'm just simplifying the graphic here. We want to include multiple genotypes or genetic strains of lice. For example, we might include one genotype that has two S alleles at a locus. We might also include a heterozygous and a homozygous R genotypes in the lice on the same farm. And we can give these, and the adults, as they reproduce, they're producing new gene frequencies, the offspring, according to the Hardy-Weinberg principle, for those interested. And we can also give the different genotypes different life history parameters. So for example, we can make lice with the S allele more susceptible to a treatment and lice with the R allele more resistant to that treatment. And the resistant individuals have a lower mortality during delousing. And of course, we now want to expand individual farms to multiple farm sites, which are occurring concurrently. And then once again, using that lice dispersal model, we can figure out how many lice larvae produced at any one farm are then transported to other farms according to their geographic location. And this whole thing is a metapopulation. So it's a sequence of different isolated populations connected with gene flow in the larval stage. So that's the model we've built. The next step is to test it out under some sort of scenario of farm selection. And for this, we simulated the evolution of azamethiphos resistance, which is one of the chemical treatments that have been used on farms. And there are a few reasons why we chose this as the first scenario to test our model with. Firstly, resistance is conferred by a single point mutation. What this means for us is that there are just 3 genotypes really in lice which determine their susceptibility to azamethiphos, which makes it pretty easy for me to code. Secondly, there's some really good bioassay data on the % survival of these 3 genotypes to azamethiphos exposure. So I can just plug these values directly into the model as new parameters. And thirdly, there's some good survey data for the frequencies of these 3 genotypes through space and through time. And so I can actually compare my model results to these survey data and see how well they match up. So I ran the model as a first little step, 537 farm sites. We ran it over a simulated 10 years. And we started off with a very low frequency of that resistant allele, which fits with the literature saying that the resistance to azamethiphos, or organophosphates, were actually present in the louse population at a low frequency before the chemicals were actually used on farms. So with this model, we can run the simulations and then produce these nice little animations. This is the coastline of Norway. Each of these dots resemble a farm site. The size of the points represent how many lice are on those farms, and the color is the frequency of the resistant allele. So we can see after 3 or 4 or so years, we have resistance emerging in orange going to red down in the southwest portion here of Norway especially, and up here. And through time, we see this kind of wave of red slowly spreading across the coastline as resistance becomes more and more frequent throughout the population. And that's going up to 10 years in now. So we see quite widespread azamethiphos resistance in a very short period of time. And if we look at the population as a whole, we see that really dramatic rise in the resistant genotype after about 4 or 5 years. And here's the number of treatments applied on farms over that period. Obviously, there's very strong seasonal variation, but we can see a gradual increase in the number of treatments applied over time. And this model really captures that key feedback, positive feedback loop, where the more treatments that are applied, the stronger the selection pressure for resistance. And as lice become more and more resistant to a treatment, you need more and more treatments to actually keep the lice under control. So something to think about is the potential of actually having some sort of mandated treatment limits on farms to prevent treatments from being used too often to accelerate the evolution of resistance. And as we saw, there was also a spatial as well as a temporal patterns in the resistance in lice. And so we can actually use these models to locate evolutionary hotspots where we can focus our monitoring and management efforts to help prevent or slow the spread of resistance. So we saw in particular the southwest of Norway here was an evolutionary hotspot. This is because of the really high density of farms in this area. So the more farms there are, the higher the connectivity. And as farms receive more and more larvae from surrounding farms, they need to treat more and more often to keep the lice infestations under control. It also means the resistant alleles can spread more easily across the farm network. And just quickly, we matched that data up with the survey data I mentioned earlier, and we did find that the gene frequencies did match quite nicely. They fell within the range we expected based on those survey data through space and through time. So the next step is exploring the different evolutionary dynamics involved. We can play around with different parameters and essentially see what happens to the louse population. For example, I was playing around with a hypothetical scenario where we've introduced a new genetically modified or gene-edited salmon strain that reduces the attachment success of new larvae by about 50%. But I also introduced a rare mutation in the louse population that has a much higher attachment rate on these new salmon strains. So there is strong selection for that different louse mutation. And as we expected from this, there was really rapid evolution of resistance in lice over a short space of time here. And the number of treatments that needed to be used in addition to the gene-edited salmon increased to help make sure the lice kept below the lice limits. But what's really interesting is that we can actually start playing around with different fitness trade-offs in the model as well. So for example, this is the same scenario as before. We have a gene-edited salmon that reduces the attachment success of larvae, but also the resistant lice, even though they have a higher survival rate on the salmon, they actually produce half as many eggs once they reach adults. And in this situation, the fitness trade-off of having lower fecundity as adults actually counterbalanced the selection for the R allele. And with this trade-off in play, the gene-edited salmon actually managed to keep the louse population under control, and also the lice remain susceptible to those salmon through time, and very few additional treatments actually needed to be applied to keep the lice levels below the lice limits. And so what do we use this new information for? The idea is we can actually play around with all these different parameters and see which ones are the most important at influencing louse evolution, and we can use these to direct the development of new technologies for louse control into the future. For example, say researchers find a number of different methods of louse control. For example, a few methods of gene editing the salmon. One method reduces the initial attachment rate of larvae. Another method affects the survival of the parasitic louse stages. And a third reduces the fecundity of the adult lice. We can see which one of these strategies are actually going to be the most effective at keeping the louse population under control at a farm network level. But it also shows us what sort of genetic traits we should be really focusing our research on the louse population as well. So we can look at what sort of traits have the biggest effect on louse evolution, what methods of inheritance might accelerate the evolution of resistance. We can also find what sort of trade-offs do we need to pinpoint so that we can actually mitigate the evolution of resistance in lice. So it's a really good first tool for figuring out where we should be focusing all our research efforts on. And the final use of this model that I'm going to get into today is how do we coordinate management strategies across farms at a regional level so that we can slow or prevent the evolution of resistance. So for example, in this scenario, randomly a quarter of the farms in the study area were assigned that gene-edited salmon, some sort of louse control strategy. And through time, we can see that even though fewer farms were using the strategy, it meant that the louse population wasn't kept under control as much in the short term. But in the long term, the evolution of resistance was a lot slower. That's because since fewer farms were using the strategy, the selective pressure was lower, and actually bided us more time before resistance actually became widespread in that population. But now what if we play around with the location of the farms, because the placement of the treated farms versus the refugia is just as important as how many farms are left as refugia. So for this example, just the other day I was just going through this and played around with what happened if only the smallest 25% of farms in the study area we used some sort of management strategy. And as we see, we saw that very familiar pattern of the evolution of resistance over a relatively short period of time. I then wanted to see what happened if the biggest 25% of farms instead used that strategy. For example, if only the biggest farms were stocking the new gene-edited salmon. We expected this to result in a much higher and faster evolution of resistance in the louse population. This is because bigger farms support a larger louse population. That means changes to the louse gene pool are more pronounced on those farms. However, surprisingly, the evolution of resistance was actually slower if only the big farms used the strategy. And that was because of, in this study area, the largest farms tended to be up further north on this part of the Norwegian coast. And these farms were more isolated, and that lower connectivity with the south in particular meant that resistance couldn't spread as rapidly throughout the study area. So essentially using these models, we can find out where the ideal spots for treated and untreated farms should be, that we can really minimize the chance of lice actually adapting to any one strategy. So what's next? We're constantly building and updating this model, trying to make it more and more comprehensive. So we want to include multiple different strategies on farms. We also want to include multiple different genotypes for resistance in the louse population, so they could become resistant to one or the other or both of the management strategies that we're using in the model. And then we can run a whole bunch of different simulations and find out which way of coordinating these different strategies have the greatest effect at controlling the louse population. For example, do we combine both strategies on all farms? Do we combine them only at some farms? Or do we completely separate the strategies through space or through time so that every farm acts as a refugium to at least one management strategy? So once again, we're looking for that perfect balance between managing lice in the short term, so controlling infestations, but also managing them in the long term, which means delaying the evolution of resistance to ensure these strategies remain effective in the many, many years to come. So hopefully I can show that evolutionary models are a really useful tool that can be used for a number of different purposes in exploring the evolutionary dynamics of lice. And I think this is really important for the current known issues of louse resistance, so pesticide resistance in Norway, but also in a lot of other regions where resistance hasn't become as widespread yet. But it can also be used as an exploratory tool to prepare us for the future and look at ways of how lice might be able to adapt to new or emerging methods of louse management, such as non-chemical controls. And more broadly, I think there is this big need for long-term goals for the sustainable louse management. And by applying evolutionary principles, we can really find new ways to safeguard the management strategies we have today to ensure they remain effective well into the future. And a big thank you to everyone involved in my project. Thank you all, and enjoy the rest of your day. Thank you.